All right, so this is kind of a story. So today we're going to talk about data handling. So we're not going to talk about any particular analysis per se, because all analyses are the same. You have data, you run tools on it. Well, of course, there are different kinds of analysis. There's different kind of data, but conceptually, tool gets input, produces analysis. Um, but I'm going to use an example today of a very simple variant calling workflow. And this workflow starts with reads. You then map these reads against some genome, and you get variants, and then in the end you get a table. It's a good scenario because it illustrates a kind of your analysis workflow when you have lots of samples. So before we get into this, essentially we're going to move through this schematics. I'm going to explain briefly. So suppose you have 4 samples, and they have forward and reverse reads. So if you're dealing with Illumina data, that's what you're going to get. If you're dealing with Oxford Nanopore or PacBio data, you don't have any paired reads there. You just have whatever your read files are. They can be in FASTQ format, they can be in FAST5, or in these BAM data sets that PacBio produces. So it doesn't matter. But paired data is a good example because it illustrates it's a layered data. So you have one sample, but it usually has 2 files associated with it, forward reads and reverse. And it's kind of a minimal example because we only have 4 samples. But the point I'm trying to make here is that very few analyses in biology are done on one sample. It rarely makes sense for 2 reasons. First of all, it's getting relatively cheap. Plus, with one sample, I mean, what can you really do with one sample? Well, if you're sequencing maybe in genome sequencing, one sample is good enough when you want to sequence a particular genome. But even then, it's better if you sequence trios. But if you're doing RNA-seq, cheap-seq, one sample is never enough, because you always need replicates and so on and so forth. So at the end of the day, you get your data to Craig Frohl here, and from him you get back a bunch of files under one drive. And then everything stops. So you can handle these files separately, but then even when you have 4 samples, that's 8 things to deal with. You know, click, 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 click. So you have to do a lot of clicking. And so what so in Galaxy we have this concept, and essentially today's workshop is solely on this concept because it's very important concept for how you do analysis and for all downstream applications. We bundle this all together, and that bundle is called dataset collection, and it's one thing. So instead of having 8 things in your interface, you just get 1. And this thing can have hierarchy to itself. So in this particular case, it's one thing. Inside that thing, we have 4 samples, and inside each of these samples, you have forwards and reverses. When we talk, so the next workshop, which will be at the end of the month, we're going to have an RNA-seq example. And in this RNA-seq example, we're also going to have hierarchy, but it will be a different hierarchy. It will have more layers because we will add different factors to it. So we'll have males, females, smoking, non-smoking, affected and affected, this kind of thing. So you can nest it as much as you like. And then that hierarchy is then understood by tools. So for example, when you run dseq2, it allows you to pick factors from that hierarchy. So it makes your life much easier. But in this particular case, so I have 8 things. I bundle it in one, and then I map the reads. So I align them against the genome. I do it in one click, but it actually starts 4 mapping jobs. If you had 100 samples, it would start 100 mapping jobs. If you had 1,000 samples, it would start 1,000 mapping jobs, and so on. So it's sort of how we split and parallelize this. And then this analysis kind of goes in that form. So we have reads, and we have map data, then we call variance. And at the end of the day, at the end here, we convert these variants because these variants of DCF format, not very pleasant to look at. We convert it to tab delimited format, but we still preserve that hierarchy of 4 samples. But then at one point, you actually need to collapse it all together, and you need to collapse it in such a way that you actually preserve the information, know which one is which. And in this presentation, we have different shades of yellow. So they're supposed to represent different samples. So in your final collapsed version of all of this, you still will be able to separate these samples. Okay, so any questions? That's the big idea here. I sent you a link, so if you check your emails, and if for some reason I don't have email of some of you and didn't send the link, let me know. But hopefully, okay. So you need to log in into Galaxy and then click this link. What this will do, it will put you in a special queue, which isolates you from all the other people who run analysis on Galaxy. And like right now, I checked, we have about 250 jobs. You don't want to compete with them. Okay, so that's the idea. I'm gonna more or less follow this tutorial. I sent a link to this tutorial in that email as well. But more or less, it's a jazz performance, not classical music. So we're gonna deviate. We're gonna interpret it. So here's our data. That's kind of the interesting thing. So let's load it up. I just need to select everything here. These are just links to 8 different data sets. Let's start uploading them, and then I'll tell you what this actually is. So I'm selecting that, going to my Galaxy. So I'm already logged in. I'm in the right queue because I clicked on that link. So now I can click on that upload button, select paste fetch, and I'm just going to paste whatever I select. But I'll click start here. So there's, all the data comes in different formats. And this is sequencing data in FASTQ format. So the problem with FASTQ format is it's a very bad format, but it is what it is, and we all use it, because there are different flavors. And there are historic reasons for that. And the flavor we all use today is called FASTQ Sanger. So to just avoid any complications in the future, let's actually tell Galaxy that. So if you start typing FASTQ, you'll actually see, wait a second. You actually see this one, FASTQSanger.gzipped. That's the data that we have here. So, if you didn't do that, Galaxy is smart enough to understand that this is gzipped faster. The only problem is that flavor of Sanger, it's hard to deduct from the data. So it's better to set it explicitly.
Can I ask a question? How would you know? Does that mean this is Sanger sequencing data?
No.
I assume this is Illumina data. So
it is Illumina data.
So you find that out by asking.
If you have the data produced in the past 5 years, it's undoubtedly going to be FASTQ Sanger data. So FASTQ is the name of the format, and Sanger is the name of the double Nobel Prize winner, Fred Sanger, who died about 5 years ago. But he didn't do anything related to FASTQ here. What this means is that so every sequencing data provides you with 2 types of information. It provides you actual basis, ATGC. It also provides you quality scores. So how good the base calling algorithm, when it was processing this Illumina picture, it's a quantitative measure of how good this call is. And this matrix, there are several ways to express that. So one of these ways is so-called Sanger matrix metric. And so this is this is the standard way we encode things right now. But if you were doing things prior to 5 years ago, if you have really old data, and that's where it gets a little complicated, because there was base 64 encoding, there was just encoding where they were just putting actual numbers in the file, so there was a lot of mess in the beginning when sequencing technologies were evolved. Anyway, I'm talking too much, so select this and just click Start. So
did you want that in a collection?
I want as a collection, but I'll do this manually. Yes, you're right. So we can create collections in many ways. One of the ways we can do this directly by uploading. Anyway, so what this is, it's this actually data from an old PNA, well, a PNAS paper we published in 2014, I think. And there was a particular family, and this family had, in this particular case, it was a mother, M117, and also there's a child, so it's mother and child. And it's data coming from either blood, which is specified by DL, or cheek, which is specified by, so it's a cheek swab or blood. So that's what this data is. But you have 4 samples here. So mother blood, mother cheek, child blood, child cheek. And each data set has forward and reverse. So yes, we can go ahead and start mapping this one by one, and we go crazy and develop this carpal tunnel syndrome. This is how it goes. With 8, you can do it. With 20, maybe you can do this too. But at some point, it will become impossible, and you will say, Galaxy sucks, and you'll never do this again. Because you really want to click 20 times or something like that. So instead, let's bundle this into a single thing. And here's how you're going to do this. We're going to click our checkbox, and select all of them. And for all selected, I'm just going to get here, which is the build list of data set pairs. You get this wizard. So it's often when you have paired M data, the data set names have this _1 and _2, which means forward, that's _1, and reverse, that's _2. So it's sort of expecting that, but let's assume that we actually don't have that. Let's unpair them all. So let's suppose Galaxy actually confused, it doesn't know what corresponds to what. So you can just, in this field, you can just enter something which allows you to tell forward reads from reverse reads. In this case, it's _1 and _2. And so it will automatically just, so you see that's mother blood forward, mother blood reverse. So it's smart enough to align them carefully. And then we can just pair them all. And in the end, that's what we're going to get. Either the samples, either forwards and reverses. And I'm going to call it something like reads, for example. And at this point, you can just click Create Collection. And what happens is that everything you had there disappears, and then you have just one thing to deal with. And that one thing, if you click on it, you will see samples. If you click on each sample, you have forward and reverse. And as we're going to talk in the subsequent, sandwiches in 519.
Yeah.
519. Across from the elevator. Okay. And again, this is just forward-reverse hierarchy, but you can have cases, controls, smoking, non-smoking, males, females, whatever. But in this case, we have this paired hierarchy. So that's a collection. That's just one thing we're going to have to deal with. And again, in this case, it has 4 pairs, but we have data, we have collections. For example, when we were doing COVID analysis with tens of thousands of samples, but they all look the same, just one thing.
So you can download out of the SRA database.
Yes.
Right? And it will default come back as collections?
Yes, so if you do that, so it will create a collection for you, yes. But
As a default.
as a default. There are some, so perhaps it's a separate conversation. This is downloading from US SRA, I mean from NCBI. There is also European SRA, NCBI. American and NCBI SRA is not reliable. So unfortunately, these things, when you have, for example, a list of, I don't know, 20,000 accessions you want to download, the chances are some of them will fail. And so we're going to have a workaround where we download them sort of individually, skipping those that are failed, and then we bundle them in the collection. But this is only, it's only going to be a problem if you have a really large list, say more than 100. Because unfortunately, it's not our problem, it's the SRA problem, is that these tools that we're using, they fail very often. European short-read archives are better, and they provide explicit links to FASTQ datasets. And NCBI SRA doesn't do that. Anyway, I'm derailing here. Okay, so let's do something with this collection. And usually, it's an old joke, but when you get data from core facility, the key existential question at this point is to pay or not to pay. And it's all the money. So when you get data from Craig Pro, data is good. But who knows, maybe you get it from somewhere else. So probably good idea to sort of figure out, is my data good? That will be an assessment of a sort. In case of Illumina data, you can do, we can use a tool which is called FastP. It's a good pre-processing tool. So I typed the FastP in the search, so here it is, and it's complaining. No FastQ or FastQ.gz datasets available. And that's because it's in this single dataset mode, but if I click on the collection mode, then it will see it. But before we run anything, we actually need to tell FastP that this is, we're dealing with paired collection here. Paired collection, that's our collection here. And one last thing I'm going to tweak here. So I'm going to click on output and set this to yes. I think in the future versions of this tool, we'll probably need to set this to yes by default. It will just create a output in a format which will then be easy to visualize. The paired collection reads JSON to yes, and let's run this. And so it starts 8 jobs automatically. So, oi, oi. Glasses. Oh. Somehow I got logged out or what? Sorry, I just cleared the cache of my browser, so there might be problems regarding. Oh, does it work for you? You don't get that error. Okay. Well, lucky me. How do I clear the cache in Chrome? Does anybody know? How do I hard clear cache? well, let's see. Let me try this again. Maybe it's all good. So the reason I'm getting this error is because before I just wanted to have the clean session, so I deleted all the cookies. It's not a good idea. Anyway, so, but it's a good it's a good example to show you how the history works, nevertheless. So the chances are, when you're gonna do analysis, you're gonna get to something like this. So it looks nasty. You have the reason that you have all these errors. And since I'm showing off here, I want to have a clean history, so I don't want any errors in my history. So provided that all this login magic with cookies worked, let me show you how to kind of move things around. So I'm gonna create a new history for myself. You don't have to repeat this. Just watch what I'm doing. So I'm just gonna create a new empty history, and then there is you can have multiple histories in Galaxy. So each history is your workspace, and so I have a bunch of them. See, not all of them are pretty. And so this is my new history, and I'm just gonna move it in here. So so this way I have the clean start, and let's call this something like all right, so let me try again. So if you're not logged in, you cannot run any tools, and that's what just happened to me.
So why did the last one fail?
Because I logged into the browser and then I reset cookies. It didn't match. The token didn't match. So you can only blame me. So the bottom line here is that it's doing this operation, and actually what FastP does, it does two things. It's a nice tool. It generates quality statistics for your data, plus it actually cleans it along. So it produces three outputs. First of all, it produces clean reads. And if you click on that, you'll see that. So you have the same structure. This collection is the same structure as that one. It produces HTML report, but the problem with this HTML report is that it's for each sample. So you can look at it, but so the problem with this report, it's beautiful and it has all the right information, but the problem is that you have to click on each and every one. And if you have 4, it's okay. But if you have 10, it's okay. But if you have 100, you're not going to be clicking that much. So this is why when I was running it, I chose to select the JSON output, because there is a really nice tool. Probably all of you know this tool. It's called MultiQC. And it's a tool which likes to take summaries of analysis, like this, and aggregate them and produce just one quality control summary. So let's use that. So first we need to tell this tool which report we want to process, and we used FastP. And it needs to know which data set. So FastP, this, and we don't need to do anything else. We'll just click execute, and this will actually give us So it will give us the stats as a collection, so there are multiple data sets there, but it will give us just a single data set with a quality matrix. And so the history design, the powers of these boxes and so on, it's going through a transformation. One of the challenges that you can see immediately here that it's not always easy to understand which one are collections and which are the individual data sets. The only thing that differentiates them here is that they have an eye icon because you can look at it, but they will have sort of different shading in colors in the next reads. So it's easier to figure out what you're actually looking at. So let's look at that. And so this is class typical fast report. This is sample data. So these data sets are small. I think each data set contains like 1,000 reads. It's easier to use for a workshop. But there's still some Illumina adapters lingering in these data. But overall, it's a really nice data set. So you're going to have to pay here. This is good data. You will not be able to make the case that facility screwed it up. Okay, so let's go back. So, and we're doing this, it's all, we're dealing with a single interface item which corresponds to, which unites 4 samples. Okay, let's do something meaningful now. So I'm gonna map these reads. Let's map them with BWA. So there are many BWAs here, but use BWM, not BWM2, not BWM2, use BWM. So these kinds of tools, I'm sure you all know what it does. It takes feeds and it maps them against the genome. But it maps them against a genome which is pre-processed. It's a special index of this genome. So in this case, it's a human data, so we can just select something like type AG38, and that's the data set. This is not the latest version of the human genome, but it's the latest, most annotated version of the human genome. The newest version of the human genome is called T2T human genome, telomere to telomere, and it's not as heavily used yet because it's not annotated well. So it doesn't have all the tracks with all the genes, expression data, regulation data, and so on it yet. It hasn't been transposed. So the G38 is the most annotated version. So this is a scenario where the Galaxy server we're using, has this data already pre-cached for you. Well, you might be working with some weird organism for which it's not stored on the particular Galaxy server that you're using. So in this case, you can simply upload this whole genome as just a data set here and use the genome from history. And what this will tell Galaxy is that take that file, make an index, and then map reads against it.
Does it save the index?
It saves index for you in your history, but it does not make it globally available.
As long as it's been done within the history.
Yes, you can always move it to another history. Ultimately, you can also, something I didn't talk about yet. There is also this concept of data libraries, and basically it's like a data folder that you can create and dump all the data there and make it always accessible. You can directly select it. So, okay, and this is the data set that I'm dealing with is a paired collection, and so it immediately sees, okay, well, that's the data I want to have. Not this, but this, because this has been processed by FastP. Do you hear me okay in the mask? Okay, so I'm loud enough. Good. So I'm not going to change anything else here. probably. And let's just run this thing. Okay, so let's start 4 BWMM jobs. So for example, when we were dealing with COVID data to analyze 1,000 samples, well, COVID genome is small, so it's easy to map. It's 1,000 samples you can process in about an hour, because individual mapping jobs are short. In this case, we're mapping 1,000 reads against the entire human genome, against the entire 3 billion base pairs. But our set of reads is small, it's just 1,000 reads, so it's not going to take that much. But it will take some 2-3 minutes. Any questions? Okay.
So the scaling on this, is it running it in parallel? So if you have like 100 short genomes, is that about the same length of time as one short genome, or are we talking about 100 times as long?
It runs 4 separate jobs. So, and they go to 4 separate nodes. So the way Galaxy is configured is that every tool has a specified destination, and so tools are different. I mean, some tools don't need much CPU, like when you count number of lines. But these tools do need CPU, and they go and they configure in such a way that they take nodes with X number of cores. I think these are configured to go to the nodes with 6 cores. So all tools have different. There's a config which tells what goes where. And we actually, so one of the interesting things that we can do is that because we were running Galaxy for the past more than 10 years, we actually accumulated enormous amount of tool usage data. And so one of the things that we can do with this data is that we can kind of train models and predict if you start a job on genome of this size with that many reads, how much time it will take. And so it's sort of an intelligent job scheduling. So given, based on our previous experience, we can say, well, this is better to send it to a node with 48 cores, or this one doesn't need much computing. So we're all competing for that queue. So what's happening here is that I have 2 gray ones and 2 yellow ones. So the yellow ones are running, and these are queued, so they're waiting. The way this workshop instance, the way this queue is configured is that every one of you can only run 2 jobs in parallel, including myself. So it's going to take a little bit of time. Well, let me tell you a little bit more about history. So, you can obviously do things. These are the operations that you can do on this entire panel. You can, for example, export tool citations. So when you write your paper, this is what you make citations for you. You can share it. The cool thing about this is you can extract workflow. And so, because what we're doing here, we're actually building analysis. We started with reads, and we ran FastP, and we ran multi-QC, and we ran BWMM. So it's a chain of events. And so workflow, so when you do this, obviously all this information is captured. And so then you can create a workflow. And workflows in Galaxy, while it's still running, if you haven't seen them before, they look like this. I'll show you something reasonably small. That's an example. It's an example of a workflow. which is sort of exported from the history. And so these are the usual tools. And if you click on every tool, you get the two interface. It's the same interface that when you're using Galaxy in normal analysis mode, it's the same interface that you get in the center pane. And the last lecture of this series is going to be API and about API's application programming interface and about contributing tools to Galaxy. So one of the things that you can do, you can run this workflow obviously through the graphical user interface. You just click run. But many people who do analysis at scale, they created a workflow, and then, for example, they want to run it on, 1,000 times. So for them, doing this through this interface is annoying. So you can do this programmatically. There is a system which can, you just give it a workflow ID, and you give it where to run it. And so workflow ID and where to run it, for example, on US instance, on European instance, or an Australian instance. So all these open public instances. So you can submit for free large analysis to them. What did I do now? Not very helpful. Oh. See, it's it appears that perhaps the node that we configured for that was not big enough. Well, let's do something about that. So I still want to do this. So let's do this. So we don't have enough memory on the nodes where these jobs are sent under umbrella of this workshop. But it's okay. So one thing about these reads. One thing about these reads is that this data is mitochondrial data. So let's simplify our life, and instead of mapping it against the entire human genome, let's map it against just the mitochondria. So let's go to NCBI. Let's just find the mitochondrial sequence. So what we need is go to genome assembly. Is this useful? Not really. I know. Here's okay.
Do you have an estimate in the memory you were using that failed your session, your previous?
So it only failed because it's a workshop queue. So if you run this outside a workshop queue, this won't fail. But yes, so if you click on the on a failed job, if you click on this I icon, I button, it will tell you, I think. Maybe I need to be an admin to see that. But essentially based on that, if you're an admin, and if you have this error, and you cannot find it, it's, well, usually what happens in this case, you just click on the bug, and you provide us with the bug report, and when we get this bug report, we know the job ID, so we can take a look at what happened. Okay, so, all right, so what I'm doing is I'm trying to find human mitochondrial genome. If you have any ideas where to find this better,
It's the mitochondrial reference genome.
all right, so exactly. Okay, So that will work. So for those of you who didn't see what I'm doing, I just went to NCBI, typed human mitochondrial genome, and as Steve pointed out, it's right in front of me, but I don't see it. So it's right here. And I'm just gonna send it to file and in the FASTA format. Let's do it this way. Actually, well, we can make it more interesting, but okay, let's not get carried away. So let's do it in FASTA format. It's okay. So that will create a file. So this will create a local file on your laptop. And then I'm going to go back to Galaxy. So I don't like these error data sets. I'm just going to delete them. Looks better. And I'm going to go here to upload data. I'm going to choose local files. And I think that's where it is. And just click start. I think it should be smart enough to figure out if this is FASTA. So sometimes you delete data sets, but you don't want them. You realize so they're actually, they disappeared, but they're still there. So you can always get them back if you want. So this also tells you how much this history occupies, and this also tells you how much in your total allocation you are. By default, we give people about 250 gigs, which is not that much. But if you need more, you email us, we'll give you more. This account that I'm using is kind of special, so you can see that I'm already using 780 gigs. What will be happening with this system in the next year is that we're going to have this concept of the scratch history. Scratch history means that you can make it as large as you want, but if you don't touch it for a week, it disappears. And so this way, this would allow people to run very large analyses and then just save whatever they need to something which is not scratch, something that will not disappear.
So when is that going to happen?
This is going to happen in 2301, so it's next release, but who knows? So actually, I don't think we don't call them by the months anymore.
No, I think it's labeled by the month when it comes.
So we're usually a few months late. So when it's January, it usually happens like in March. So because we're fixing things. Okay, so we're back to the where we started. So we have that sequence now, and that sequence is. So let's run BWAMM again. So I'm going to run BWAMM. But now I'm going to, so it's a good exercise in there. Let's use the genome from the history. And it will ask us, okay, which one? So here it is. It's going to be paired collection. That's the right data set. So use genome from the history, specify the FASTA file we just downloaded. It's a paired collection, and that's the data. And now we pray. This is. So now it's a two-step process. It makes the index first, and then maps to the index. Will succeed. Okay, so now we have 4 BAM data sets. And these are binary data sets. You cannot really take a look at them. So this is map data. And in that form, you can do many things with it. You can covariance, find, cheap seq peaks. If you're doing a genome-based RNA-seq, you can do RNA-seq, but although usually when you're interested in gene expression, you're actually not mapping against genome, but you do pseudo-alignment against actual transcripts. So there's a different tool for that. Maybe you heard of these tools. It's tools like Callisto or Salmon, these kinds of tools. But right now, I'm going to covariance. And how did I do this in the tutorial? In the tutorial, okay, in the tutorial I did this with low freak. Okay. So let's use low freak. Let's start running, and then I'll explain what different things with different variant colors do. So low freak, it's actually a family of tools. If we were doing this to get our publication, you actually need to prepare data a little bit. You need to do a bit of a realignment and quality recomputing, but we're not going to do this right now. We're just going to go straight to covariance. So again, we need to click on the collection so the tool sees the data. So here is the data set, blah, blah, blah. And it needs to know what the reference genome is. And just like BWA, it can be either locally cached, or we can provide one from the history. But in this case, we do have one in the history, so we're going to provide it. And let's go close for variance, and then so several modes. You can call only SNPs, or you can call SNPs and indels. Let's just call only SNPs in this case. And I'm not going to modify any defaults. Let's just run this. But the variant calling is surprisingly so. If you look how money are spent in biomedical research, in biological research, of course, most money is spent on human, because that's because of the biomedical driver, because NIH funds a lot of things. Problem with human is that it's deployed. I mean, it's not a problem, it's just the way it is. So it's deployed. And so when you have deployed organism, it's relatively easy if you have large coverage. You can have, at any given position, you can have homozygote just like in reference, so it's the same base as in reference. Or you can have, alternative homozygote, a position where you have a different base, and it's uniform across all reads. Or you can have a heterozygote, where you have, for example, reference base from father and alternative base from mother. But you have this deployed expectation where you have either 200% or you have 50-50. It can get complicated to figure out 50-50 if you have low coverage, because when you have 4 reads and one of them is A and 3 of them are T, it can be a heterozygote, but it can also be a sequencing error. But however, in this case, we are calling variants in mitochondria. Mitochondria is not deployed. every cell has lots of mitochondria, and each mitochondria has lots of copies of its circular genome. So it's like calling variants in E. coli or in COVID. We don't have deployed expectation here, which means that we can have alternative allele frequencies anywhere between 0 and 1. It's a separate story how we reliably call variants, because, for example, with Illumina, this is Illumina data, you cannot really call anything below 2%. It's just, you cannot trust, you have enough noise to obscure anything below that frequency threshold. But I'm saying all this because low freq is one of the variant callers specifically designed for this case. There are some other variant callers which can be used for both scenarios. For example, FreeBase or some variant caller from GATK toolkit, and they have germline and somatic modes. And somatic mode is kind of similar to this scenario, because in somatic mode you have a bunch of cells, and most of them are referenced, but some of them are mutated. For example, you have one cancerous cell in some constellation of cells. So they will also have these frequencies between 0 and 1. There's no deployed expectation there. But this particular variant, so this is what we use, for example, for COVID. Okay, too much talking. So, so we're kind of progressing through this. We're at that stage right now. And so it's about 1 hour, so let's make a 5-minute break. So get water, finish your sandwich. If anybody wants coffee, I can make you coffee. Anybody wants coffee? Well, I want coffee. So, okay. So right now it's 2:58. Let's get back at 3:05. So I'll restart at 3:05. If you have any questions, I'll get coffee. I'll get here. You can ask me questions. By the way, I failed to introduce Greg Van Kooster, and he's been with the team for quite some time, and right now he's running Penn State instance of Galaxy. So we are using this usegalaxy.org, which is a big public site, but there's actually Penn State instance dedicated to Penn State, and it has much better waiting times than the public service. So it just doesn't have a training key. But he'll tell you more about that. So because actually we organized this workshop to kind of popularize actually Penn State instance, but I'm just doing training on the public instance because at this point it has more capacity. But it actually runs also in Texas at the same computing center where the main Galaxy is. Do you have any questions so far? That means I'm very clear. While we have this, let me tell you one thing I'm really excited about. It's, let's see. Oh, God. So it's a scary diagram. But what this is a set of workflows that we're using for assembly of really large genomes. So mammalian scale type of genomes. And so each of these boxes is actually an individual workflow. And so we have several nodes. This data actually, it's not based on Illumina at all. So there are some Illumina data sets, but there, when you have a trio, you have the individual you're sequencing plus its mother and father. You can have mother-father data as Illumina, because what you need is you just need the paternal and maternal k-mers, for which Illumina data works very well. And Hi-Fi, this is spec bioreads, and Hi-C, it's also Illumina data. This is it's kind of like really long insert paired end data. So it shows, it defines which regions of the genome are adjacent to the same linkage in the same chromosome. And that's sort of a decision tree. If you're rich enough to generate that data, then you're using these trio workflows. And if you only have solo data, then you're using solo workflows. And you go through this. And in the end, you're actually getting really nice, really good quality assembly. And we assembled 32 VGP genomes using that, from fishes to lemurs and things like that. And it's at this stage, this is actually the assembly part, which uses Hi-Fi to do like bioassembly. That's the computationally intensive step. And for this, so for example, PWA jobs, which we were just running, they go to a node with 6 cores. So these jobs go to nodes with 128 cores and about half to 3/4 of terabyte RAM. But it's still remarkably efficient, because you could not assemble human-sized genomes on a single node 3-4 years ago. Now it's possible because of the long reads. But before, you needed a big, large shared memory cluster, but now it's possible. So it's really, so this is actually interesting for the giraffe work and for what we're doing with Drosophila. So anybody who does large genome assembly, you can talk to me later if you're interested. Okay. Well, let's continue. All right. So,
So will we continue to have access to this training tutorial page?
of course,
Okay.
they're public. They're not. There's nothing. Everything we have is open. And on top of this, I'm actually recording this. So if any of you really wants to listen for, if you can't get enough of my accent, then I'll provide you opportunity to listen to it again. So anyway, there are some people who are not here and ask me to record some recordings. So I'll edit it a little bit. So I'll cut the break from it, for example. But I'm not going to edit it much. Do I have enough battery, by the way? Yes. Okay. So we call it variance. This is data we got. It looks very clean, actually. So in this particular case, well, you have some variance. That's the reference base. That's alternative base. The living frequency is 99. So it's really not. It's not a variant at all. It's probably just a sequencing. So, I mean, it is a variant, but it's not a heterozygote. It's clearly a variant. It's clearly different from the reference, because the reference has a T, this has a C. So it has a low frequency is essentially 1. So there's no room for. It's possible that it's a mixture, but with that percentage, it's not really. Not really. So now imagine we did it on 1,000 samples. So you're going to get 1,000 data sets like this. And this is VCF data. It's good for some things. For example, it's good if you're going to feed these variants to some downstream workflows for recalibration, and there are many tools which accept VCF. But in this analysis, I just want to know, do I have any differences between child and mother? That's all I want to know. But now just imagine that you're analyzing not one family, but for example, 100 families. The PNAS paper, which we published, I think had 100-something families. So you're going to have lots of these. And this is not a good thing to look at. So the first thing I'm going to do, I'm going to convert it into tab delimited format, because tab delimited format is easy to deal with. So, and if you go back to training, it actually sort of explains how to do this. And I'm again doing this stupid thing I forgot. So for those of you who were here last time, I'm doing this stupid thing because I have training open in one tab of the browser, and I have Galaxy opened in the other tab of browser. So I'm going back and forth. But actually, if you click on this hat and go to and find this tutorial, and in order to find this tutorial, it's the tutorial I'm using. It's in Galaxy tips and tricks. So it's in using Galaxy and managing your data, and it's just using data set collections. So essentially, I now see it from within Galaxy, and the beauty of this is that well, it's an older tutorial. No, it's all good. So the beauty is that all the tools are these blue buttons, and so you can, for example, what I was doing, I was searching for tools like snipf. That's one way of doing this, of course. But if I go directly from the tutorial, I'll just click on that button, and it brings up two for me. So I don't need to do any searching. This is a really cool integration between tutorial and actual interface. So, all right. The one thing here is that I need to select, I can never remember this, but I actually need to, this is the fields, the values that I want to extract from the CF file. So the way I'm doing this is I went to the tutorial, got to this step, and I'm just carefully selecting this string from Chrome to GP4. Just make sure you don't select any axes. Okay, and I'm going to go back here, and if you don't want to do this, it's basically chromosome. Well, we only have one chromosome in this because we mapped against mitochondria. So chromosome, in other words, which sequence we mapped against. We mapped against one sequence. So it's going to be the position, so where our variant is. What's the reference allele? What is the base in our reference genome? What we're actually calling? What's the alternative? This is the call quality. And so these are the interesting fields for our particular analysis. This is depth, so how many reads at that position, allele frequency, and there's also two metrics which are there. This is where all variant call is different from each other, because if you run GATK, you get one set of fields. If you run FreeBase, you get another set of fields. If you run low-freq, you get another set of fields. SD stands for strand bias, and GP4 stands for depth. There are four values. How many reads on the plus strand support reference allele? How many reads on the minus strand support reference allele? How many reads on the plus strand support alternative allele? And how many reads on the minus strand support alternative allele? And strand bias in these cases is it's a very important metric, because if you have a good variant, it should be called equally well from both strands. You have to be careful when you're doing, sometimes you do variant calling from RNA-seq data. And RNA-seq data can be stranded. You have stranded RNA-seq protocols. So in this case, you're going to have calls that will be biased. But they're not biased because they're bad. They're biased because of your protocols. You need to know what your protocol is. Okay, so I'm just selecting these, and I choose as input the variant, which we just called, and I'm just clicking execute here, I think. Or did I miss something? Yes, I did miss something. Should have selected one effect for a G. So let's do this again then. So I'm gonna one effect per line means you can have a site where you have two alternative alleles. Say reference is A, but you have T and C. And if you don't select that, then they're both will be reported in one line of receive. But if you separate them, they'll be in separate lines. And it's easier when they're in separate lines. So what we're going to do next is this collapse. After we ran this tool, SnpF field extraction, it generates a collection with 4 tab delimited files, so variants in tab delimited format for 4 samples. But ultimately, I want to dump it in a single data set. So I can load it, for example, I don't know, if I like Excel, I can export it to Excel. But I don't like Excel, so I can use it in Jupyter or in Pandas or in R data frames and so on. But you don't want 4 data frames, you just want 1 data frame. Again, we only have 4 samples. If we have 10,000 samples, you don't want to deal with 10,000 data sets. So it's done, and if we look at this, well, these are just tab delimited data sets. So probably there are variants. I just, in order to know, I just need to click on all 4 of them. I don't want to do that. And so here we come to this important set of tools. They're called collection operations. These are tools designed specifically for dealing with this agglomeration of samples. So for dealing with collections. And there are many of them, and we're gonna go through them. But the one I'm going to use right now is called collapse collection. And I think I have a good illustration of what it does probably here. Yeah. So it sort of has 3 modes. So suppose I have a collection, and each element in the collection, well, if you go here, so these things, they have names. These names are derived from the files. But let's suppose that the names are 1, 2, 3, 4. If I concatenate them all, then I'm going to lose all the information which is what, obviously. So there are kind of 3 things that I can do. I can prepend the sample ID to each row in the table, or I can do, for example, if I output FASTA files, I can, for example, just print an extra line above each block, or so there are different ways in which I can do this. But the reason, what I'm interested in right now is doing this. So I merge them all together, but I add a separate column which provides identifier. So then when I load it to Excel or Jupyter, to Pandas or R or whatever else it will be, I know what is what. So this is the collection I want to run this on is, I have 2 of them because I ran this twice, but maybe you have just 1, but The collection I want to run is snip extract fields. And these files, they have headers. So they have chromosome position, and so I can say here that yes, I want to keep one header line. Obviously, if I concatenate 4 files just like that, it will have 4 header lines. I don't need that. I just need one. And I want to prepare file name, and it's same line in each line in the data set. It's this option B, option A. And I just run it. There's another tool which has similar power, and it's called column join. And so in this case, suppose that you have collection with 3 samples. This is sample 1, this is sample 2, this is actual data in these files. So it has some ID column and some values. And I want to kind of join them together on some given column. For example, I want to join them together using this column as the common field. So it's like a relational join. Actually it's like relational left join in this case. So what will happen if I run this tool? It will give me a single data set which will have unique values of this column in which I'm joining. And so for example, in 1, I have A in blue data set, and I have A in yellow data set. So they're here, and in green I don't have anything, so I'm putting a dot there, or some other symbol you can put. I don't know, in R it's probably NANs or something like that. So you can merge them this way. So you add column for each data set. You can do this on 1,000 data sets. This is going to be a data set with 1,000 columns, but then you can transpose it perhaps. In the meantime, it's done. And so what do we see? So we don't see actually. Well, we might have one variant with frequency of 4%, and that's actually probably a good variant, although not really. So, okay. I'm just looking at the frequency column here. So most of them are fixed. So most of them are 1s. 0.99 I would count as 1. So this one, maybe there is something there, and this is child cheek sample, so it's different tissue from blood. But these are the counts for, these are the strand-specific counts. So I have 89 references in plus, 89 references in 7 references in minus, 4 alternatives on plus, and 0 alternative on minus. So it's a corner case. I don't know, but it's not a good call because I have the strand bias here. The counts are so low that it might be the real thing. And actually, in the real data set, because I only sampled 1,000 reads here, it is a variant at this position. So there is a difference between child and mother at this particular site. But because I have a subsample of reads, and it's sort of a corner case, I don't know whether it's a good variant or not.
So when you ran this, did you allow for heterozygosity or heteroplasmy in this case?
It's a non. I mean, it's a mixture type variant caller. So
so it would allow that.
It allows by default.
And just so I understand, so this variant caller is actually calling variants relative to the reference genome.
Well, they all do it this way. Yes. So it's all aligned against human mitochondria, and that's the differences that it found. So in most cases, it's figures differences, which is normal. They all have different mitochondria, and they all they're all, well, actually, so let's see. Now, in principle, a child should have the same frequencies at these sites, and they do. So it's sites 152 and 16519, so they all have them. But that's the new one. This one, mothers don't have it, and the other T-shirt child don't have it. So, okay.
But if, in this case, I mean, if it's mitochondrial DNA, isn't it possible you could have a low frequency heteroplasmy?
Of course, that's what it is in reality. Yes, and it's if I remember correctly from that paper, it's in one of the tRNA genes, so it's a very bad mutation, and if it goes, but it has low frequency here. So a lot of mitochondrial mutations, they have frequency dependent phenotype, and so in this case, the child is normal, but if you get 10% and up, you start having serious issues, and because mitochondrial inheritance is sort of unpredictable because of the bottleneck during the oocytes are produced, then actually the study was about that. Can we find these differences between offsprings and parents in these pedigrees? And mitochondria is maternally transmitted, so we don't care about fathers here. They're not that important anyway. So, okay, so actually I think I'm running ahead of time. So let's do something else with it. Let's go beyond the tutorial, unless you have any questions. There's many operations in the collection. You can sort them. So sometimes, I guess I'm trying to find an example. Sometimes you have two collections which represent same samples but different types of data. I don't know, for example, one collection contains genome sequences for 10 samples, and another collection contains annotations for these 10 samples. And you want to do something on them together. So for example, you want for each genome, given that set of annotations, extract sequences which correspond to coordinates in this annotation file. So you can do this using collections, because what Galaxy assumes is that first element in this collection corresponds to first element in that collection, so it slices them that way. But obviously, in order to do that, you need to make sure that they're sorted in the same way. So it's really the first element of that corresponds to the first element from the other one. So this is, for example, what sort collection does. You can rename them. So there's many things, and this tutorial actually describes all of them with examples.